Okay, guys, so just to continue on from the sugar phosphate backbone, what we're going to do is we're just going to think about base pairing. So I've already mentioned we did this in Nat 5, but just as a quick reminder, you can see in this diagram that G, or guanine, always base pairs with C, cytosine, and T, which is thymine, always base pairs with A, which is adenine. Okay? And as it says here, what bonds these together is weak hydrogen bonds. So between here, joining these two bases, is weak hydrogen bonds. Okay, and later on we will be learning about why it's important that these are weak hydrogen bonds, because it means that they can separate quite easily. Okay, in the process of DNA replication, but that's something we'll come on to later on in the course. Okay. So as I said, yep, A with T and G with C. And as I reminded you, you can see that the curvy letters C and G, both curvy letters, they go together. So that's an easy way to remember it. Whereas capital A, capital T are quite straight letters and they go together as well. So that's just a quick way to remember it. Okay, if you're ever in doubt. Okay, this next part is definitely new. This is not something that we did in Nat 5. Okay, and this is, we're going to speak about antiparallel strands. Okay, so if things are parallel, it means that they go in the same direction, okay, side by side. So a good example would be train tracks. Okay, they run parallel with each other. Okay, the tracks of a train. Antiparallel, as the name suggests that they still run side by side, but they are going in different directions. Okay, and that is what a DNA molecule looks like. So if you have a look here, you can see that although they look similar on either side, this one appears upside down, and that's because this DNA strand is going in this direction, and this DNA strand is going in this direction. So although they're parallel, they're going in different directions, so we say that they are antiparallel. Okay. Now, this part here, it says one strand has the three end. So when you see this little, what looks like an apostrophe, okay, we say that's the three end, okay, at the bottom of the strand, and the other has the five end at the bottom. What does that mean? Well, this is actually a little bit of extra work, but let's just have a look here. So here we have our deoxyribose sugar, okay, and at each point of this shape, okay, there's five sides. This is a pentagon. Okay, so each corner of the pentagon, you can see there is a number. Okay, and this is to do with basically the number of carbon atoms. Okay, so this is the first carbon. This is the second carbon. This is the third. This is the fourth. Okay, and this is the fifth up here. So these numbers are relevant because they basically tell you which number of carbon is at the bottom of the strand. Okay, so for example, here you can see this is telling us that down here is the three end of the strand and up here is at the five end. That tells you that the five end is at the top, whereas the three end is at the bottom of each molecule. You see that? Of each nucleotide, sorry. Again, five end is at the top, three end is at the bottom. Here, five end this side, three end is at the bottom. Okay, so this runs from a five end to the three end. This one is the opposite. You can see up here, because it's been reversed, the three end is now at the top and the five end is at the bottom. Here, the three end is at the top, the five end is at the bottom. You can just see that's what we mean by antiparallel. Okay, now this is a process that sometimes it's just good to practice some questions on it. So there are some questions in your booklet on this, and hopefully that will help our understanding of this process. Okay. Okay. Now, when we do go over DNA replication, this will become very relevant. Okay. So again, this will hopefully enhance your understanding of this process. We're just doing an introduction at the moment. So as it says, nucleotides are only able to join the three end of a strand. So when you build a DNA strand, okay, you obviously add nucleotides together. If you were building a house, you don't start with a roof; you have to build up. Okay, so you brick on top of brick on top of brick. Okay, and that's the same here: nucleotide on top of nucleotide on top of nucleotide.